Thanks all for coming. So it's my great pleasure to introduce Professor Pierre from UC Irvine. He's currently Chancellor Professor in the Department of Computer Science, Director of the Institute for Genomics and Bioinformatics, and Associate Director of the Center for Machine Learning and Intelligent Systems at UC Irvine, as well as the long-term focus of Pierre's research is about understanding intelligence in brains and machines. And so he has made several contributions to the theory of deep learning and developed applied deep learning methods for problems in natural sciences, as well as Pierre has a very famous book on bioinformatics. I think early people who started the topic of bioinformatics. So Pierre, thank you so much for coming to Hong Kong.
Well, thank you very much, Steve and Indica, for the invitation and the great hospitality. It's a really pleasure to be here. And today, my talk has two parts. I'm not sure I'll ever get to the second one, but the first part is on circadian rhythms. Most of my talk is going to be on circadian rhythms. And if there is time, I'll also say a few things about the theory I'm trying to develop about neural network capacity. I was not sure about the format of this workshop. I know it was about biology and some computational methods, so these are the topics that I chose. So circadian rhythms are quite interesting, and most of the time you don't think about circadian rhythms. The only time you detect them is when you travel and you have jet lag. That's when you think, oh, something is going on about my rhythms. But in fact, they are extremely pervasive and they completely control your life. So if you record the activity of mice, these are traces recording the activity of mice during the day and during the night. Mice are nocturnal. You see that there is a lot of activity and movement during the night, and then during the day, nothing, right? Well, if you do the same thing on human beings, you roughly see the same thing. So every day you wake up, you eat, you go to work, etc., you eat, and then you go to sleep. And you repeat this every day of your life like a complete robot, right? You're a circadian machine. So there is something very, very strong about circadian rhythm. They completely control your life. But in a certain way, they're also flexible, and that's why you're not too rebellious. Because if you want to watch a movie one evening or skip a meal, nothing seems to happen. So they are very strong, but also very flexible, and this is quite interesting. And the purpose of my talk is to dig more deeply into this phenomena and to look at circadian rhythms at the molecular level. What is going on in the proteins, in the gene expression, in the metabolites, etc., in your body throughout the day and throughout the night cycle. Of course, there are other rhythms, there are seasonal rhythms, etc., but by far the circadian rhythms, the 24-hour rhythmicity, is the most prevalent and interesting. The first to study circadian rhythm were actually biologists. Basically, they noticed that plants tend to open during the day, follow the sun, and then they close at night, and so they thought, what happens if you put a plant in a closet? And what happens is that it keeps opening and closing with a 24-hour rhythmicity, at least for a few days. Then it dies, of course, because it doesn't have any light. Okay? So if the plant keeps opening and closing, there must be some kind of clock inside the plant, right? Because it doesn't receive any light signals in the closet. And so we're going to try to find out where is the clock inside the plant or inside the mouse. Linné, for instance, noticed that different plants tend to open with slightly different phases. So he came up with this idea of the Linnaeus garden, where he could tell the time by planting different types of flowers. He could tell the time of the day just by looking at which flower was open at a given point of time. So this is all historical anecdotal evidence. And then in the 60s and 70s, this crazy person, Michel Siff, is a Frenchman, used to go in caves where, of course, there is no light, etc., with all kinds of wires attached to him, to study circadian rhythms, to see what happens if you are deprived from light and just living in a cave, etc. And a number of interesting observations were made at the time. For instance, the periodicity is there, but tends to increase a little bit, 25 hours, 26 hours periodicity. After 30 days, he became quite depressed, etc. But again, all this is very macroscopic, and what we're going to do today is to go to the microscopic level, look at, again, molecules in different organisms and in different tissues. It's also known, and this is also a macroscopic observation, that there is a nucleus in the brain, in the hypothalamus, called the SCN, the suprachiasmatic nucleus, which for a while was believed to be sort of the center of circadian rhythms. It has about 30,000 neurons that are firing very rhythmically. It receives directly input from the retina, so there is light information coming directly to this area. And it secretes hormones that go to the rest of the body. So it definitely plays a very important role in circadian rhythms. But again, as I will show you, if you look at a cell in the liver, you will find rhythmicity in that cell, regardless of whether there is the signal coming from the SCN or not. So how do we study circadian rhythms at the molecular level? Well, today we have all kinds of techniques for looking at gene expression, for instance, using DNA microarrays, or now using so-called RNA-seq. So you can sequence the messenger RNAs that are produced in a given cell. So you can interrogate gene expression at a given time point, and then you can do the same experiment 2 hours later or 4 hours later, and repeat this in a systematic fashion over 24 hours or 48 hours. And so these are examples, for instance, of gene expression experiment. These are different genes. One is called clock, one is called FGD6, for instance. And you can measure them, again, it's expensive, but typically it's done every 4 hours, over 24 or 48 hours. And you can see, for example, that this guy seems to be rhythmic. The blue trace, so these are experiments done in mice liver under different diets. The blue trace is for high-fat diet, and the orange trace is for a normal diet. So you can see that, for instance, for this particular gene, in both cases, under both diets, you see a 24-hour rhythmicity from this trace, but you can see that there is a shift, a phase shift that seems to be present in the mice fed with a high-fat diet. And this trace is not periodic, so you can get a lot. You have about 20,000 genes in mice, and you can get the trace for all of them, and you can look at which one are periodic under certain conditions, etc., etc. So that's one set of techniques, high-throughput omic techniques for interrogating gene expression. But you can do the same thing for protein, so you can measure protein's level if you want. It's a little bit more tricky and expensive, but it can be done. And you can also measure metabolites, which are small molecules that may interact with proteins and other things. You can measure thousands of metabolites with current methods using mass spectrometry. And again, you can do it in a systematic fashion at different points in time so that you can get these traces. Okay? Any questions so far? All right. So when you do that, what do you see? You see that about 10%, I'll talk mostly about gene expression, so let's talk about transcript primarily, but you see the same thing with metabolites or with proteins. Roughly 10% of the transcript are oscillating in a circadian fashion, no matter which experiment you do, whether you do it in the liver, in muscle tissue, in brown fat, in the brain, in the SCN, in the hippocampus. All those experiments have been done. You typically find 10% of the transcript, which is about 2,000 of them, are oscillating in a circadian fashion. It's 24-hour rhythmicity. And the interesting thing is that when you do a perturbation, for instance, you can do a genetic change, you can knock out a gene, any gene, you can have a change in the environment, a change in the diet. We're looking at mice under exercise conditions versus no exercise at all, etc., etc. You can look at different organs, different tissues. It doesn't matter, you still see this 10% oscillating, but across different experiments, it's not the same 10%. It's a completely different 10% with very little overlap. So you see 3 examples here of 3 experiments. These are the number of things that are oscillating, and you can see that intersection is fairly small in these cases. And if you were to look at what is common to these different intersections, you would find a very small number of genes that I'm going to show you. Okay? So the interesting finding is that pretty much anywhere you look at, you will find that 10% of the molecules present in a cell are oscillating in a circadian fashion. But when you look at different cells, different conditions, etc., you see a lot of variety, very little overlap. Okay? So what is the core that is common to all these intersections? Well, you find something called the core clock, which is described here, which is a sort of molecular circuit. It's a very interesting negative feedback loop that you have in any one of your cells, which is really at the center of circadian rhythm. So what happens is that you have 2 genes called clock and BMAL that produce the corresponding proteins. They dimerize and they bind to the promoter regions of a number of genes, but in particular genes in the cry and in the per family. So cry1, cry2, per1, per2, etc. When they bind to the promoter, these proteins are expressed, so the cry and per proteins are produced. And these proteins do a bunch of things, but ultimately they form a complex that comes back into the nucleus of the cell and binds to the promoter of clock and BMAL and inhibits the production of clock and BMAL. So you have a typical transcription translation negative feedback loop here, which has this almost perfect 24-hour periodicity. Okay? A lot of components of this loop are well understood, but not everything. For instance, the complex made by the cry and per genes is not completely understood even today. Nobel Prize for biology, the last one, was for the discovery of per2 action. So that's called the core clock. It's what is present in the intersection of all those Venn diagrams I showed you. But of course, there's a lot of other things that are oscillating, right? In a typical experiment, I told you 10% of transcripts are oscillating. So you have 2,000 genes or transcripts that are oscillating. In this picture, you only have 15 of them, right? It's a very small subset. So there is much more, yes?
Sorry, can I interrupt you?
Of course.
For the previous slide in the Venn diagram, the interception, I'm a little bit confused. Interception just means the gene, they will still be expressed, right?
These are the genes that are oscillating both in the wild type and in the clock knockout.
Oh, so not just
Okay.
expression, it's oscillate.
Yes, this is all oscillation. Yeah, there is 20,000 genes. What you see in these pie charts are on the order of 2,000 genes. Of course, there is a lot of variability, so it's biology. So you have, for instance, 1,156 genes that seem to be oscillating in a circadian way in the liver of this wild type mice. And if you knock out a particular gene, only 238 are oscillating. Well, 238 plus 404 are oscillating. And in the intersection, you have only 404 in this particular experiment. This is the same thing, but now you change the diet, etc., etc. We have built a database that has 250 such experiments. So I'll show you the results of what you get when you combine all this data. Okay? And the core clock that I described to you, which is about 15 genes, is what tends to be in all these intersections across all these experiments. You can discover it in that way once you have all the data. Okay? Yes, another question.
Like how many of them are like the household genes?
What do you mean by
Housekeeping genes.
housekeeping genes? Those in general do not oscillate that much. There are a few genes that tend not to oscillate. I'll come back to that.
So most of them are not housekeeping genes, but like more like the time clock quarter.
I'll come back to that. In fact, almost any gene, we find 95% of genes can oscillate at least in one experiment. But I'll get there. Now, one reason for studying circadian rhythms, of course, is clinical applications in medicine. So there is a lot of evidence that there are diseases that are associated with perturbation, disruption in your circadian rhythms, depression, obesity, aging, etc., etc. So to better understand diseases, there are good reasons for better understanding circadian rhythms and how they can be perturbed. But the other side of this also is the intervention side. So what you have in this table are a list of drugs with their sort of market size, their indications, and then the genes that they target. And many of the genes that are targeted by the drugs are circadian. So the corresponding genes and proteins are oscillating. So it would be nice to know what is the optimal time at which one of these drugs should be taken to optimize its effect, right? Right now you're told to take a drug in the morning or in the evening. It's a very vague indication. And there's a movement towards precision medicine. Ideally, you would like to know what is the best timing for taking a certain drug. And the best timing may be different between two individuals. And if you are able to give the drug with an optimal schedule, you may be able to give a smaller amount of the drug, which also reduces side effects, etc. So that is another reason for studying these rhythms. Okay, so how do we do that? Well, the first technical problem you have is you get these measurements. You get 20,000 genes measured every 4 hours over a period of 24 hours or 48 hours. So it's a relatively sparsely noisy trace. You have to determine whether it is periodic or not. If it's periodic, you have to extract the amplitude, the phase, etc. And there is a number of programs and techniques for doing that. We use deep learning because we found that that's where it works the best. So basically, we put the time series as the input to a neural network, and in the output, we either predict whether it's periodic or not, or derive its amplitude, its phase, etc. What is interesting from a machine learning standpoint in this case is that you can train this system with a lot of artificial data, actually, because you can just generate traces like this using mathematical functions. You subsample them every 4 hours, and then you use that for training the system, in addition to data from biological data. You may think that Fourier methods should do very well on these kinds of problems, but actually they don't work very well because the data is the sampling is sparse and it's very, very noisy data. So all the programs that are used for doing this, and there's 2 or 3 out there, they don't use Fourier methods. Another program that we have is a program that takes gene expression taken at a given time point and try to infer at what time that expression was taken. One reason for doing that is that in the last 20 years, a lot of experiments were done at a single time point. People were looking at, say, liver cells with a drug or without the drug, and they did the experiment at a single time point. Of course, if you know that there is all these oscillations, you realize that taking measurements at a single time point may not be very meaningful. And so if you want to go back to past experiments, you would like to attach a timestamp to those experiments. Not always, the information about the time at which the experiment was done is available. So to do that, we were training what is called an autoencoder in machine learning. So a system, again, a neural network with multiple layers where you have a bottleneck with essentially a sine and cosine function that are trying to predict the phase, which is essentially the time, of the input measure. So we're using this, we're training the system to try to impute the time of experiments that were done in a non-circadian fashion at the signal time point. Okay, so we have done a lot of experiments with our main collaborator, Paolo Sassone Corsi, who is running a biology lab that is focused on circadian rhythms. So these are examples of some of the papers that we have published together, but they all have the same flavors. These experiments are done on mice, circadian experiments, and you compare two conditions. Typically, Paolo is interested in diets, so we have looked at high-fat diet versus regular diet, ketogenic diet. We've looked at fasting. There's a lot of interest in fasting these days, and we're looking at exercise, etc., etc. And other labs have done similar things. And again, you can do this in the liver, in liver tissues, or other tissues. And as I said, we have aggregated by now, collecting data over the web and from Paolo's lab, about 250 such data sets. And yes, of course.
In the liver, your cells are proliferating, right? Liver cells. So when you study liver cells, so how the cell cycle? And
It's very important. The cell cycle and circadian rhythms are very connected. I'll come back to that.
okay.
I'm skipping some technical details because, again, when you say liver, you can do single cell. That's one way to do it. A lot of these experiments are not done single cell, so you get a sort of mixtures of cell, etc. And you can do both. But it's a very good point, and I'll come back to the cell cycle. So this is an example of what you get when you aggregate a number of experiments. So maybe there is only 20 here, different experiments. And again, you look at these intersections. So this is the number of genes that are oscillating in each one of these experiments. So you see it's roughly 2,000 plus or minus 500, right? That's the 10% I was telling you. And these are the intersections that you find across pairwise experiments, and they are relatively small. And so if you aggregate all this data and you say, if you take the union, how many of the transcriptome, what's the fraction of the transcriptome that is capable of oscillating in at least one experiment? In this case, you found 68%, which is already a fairly large number. And when you go up to the 250 experiments that I told you that we have now, we get 95%. Okay? So that's the first very striking finding, which says that almost any gene is capable of producing transcript that can oscillate in a circadian fashion under at least some set of conditions. Okay? Genetic, epigenetic, or environmental. Okay? And you can do the same thing with metabolites. That's the same experiment done with metabolites, and you see roughly the same thing. In each experiment, 10% of metabolites tend to oscillate, and if you aggregate things across experiments, you get very large fractions. The next thing you can do when you have 250 such experiments, you can just build a simple histogram of what are the transcripts that are found to oscillate in the largest number of experiments, 70 out of 250, etc., etc. And immediately you rediscover the clock automatically. Okay? All the blue bars corresponds to genes that are in the core clock, BMALPER, CRY, etc. And you can see that these tend to oscillate in most experiments. Not in all of them, but in most of them. You also discover that there are RNA binding proteins, RBP, that are very high, that tend to oscillate. So there is an important interaction between circadian rhythms and RNA binding proteins. And then, of course, we look at transcription factors, and there is all kinds of transcription factors that are playing a very important roles and that are circadian in a large number of experiments. Okay? And some of this was not known, so just by doing these simple statistics, you discover interesting biology. And these results were done with mouse data. We have done the same thing now with baboon. This is baboon, and you see similar things. Okay? So, to summarize what I told you so far is that there is a core clock that contains a dozen genes, but if you look at any tissue or condition, you find that 10%, roughly 10% of the genes, of the transcripts are oscillating. And if you look at intersection between two different experiments, you find a relatively small intersection on the order of 2%. And if you take at the union, you get almost anything, which is about 95%. And you see that every time there is a change, whether it's genetic, epigenetic, or environment, there is massive change in oscillations. Completely different sets of molecules, of transcripts are oscillating. There is changes in amplitude, there is changes in phases, etc. And so the question is, how can this be possible? Because if you think of cells, of tissues as dynamical system, these properties are quite strange and require a framework for trying to explain them. And so I'm going to describe the framework for trying to explain these interesting observations. So the first thing you have to realize is that if you look at the molecular species, a transcript, it cannot oscillate in isolation, right? It's not possible to have the concentration of something going up and down in a rhythmic fashion just by itself. So what you have, of course, in the cell is a lot of complex feedback loop. I showed you the clock, but there is many more, where you have transcription factors activating particular genes. You may have protein-protein interactions, etc., feeding back into the original transcription factor. And so what really oscillates are these loops, right? And in a cell, you can build a circuit. I mean, there's various ways of building these diagrams of molecular interactions in cells. But of course, we know there is many such loops. Hundreds of thousands, if not millions, of possible loops inside a cell. So that's good news. It means you have a lot of potential oscillators inside the cell because you have all these loops. That's good. But then the question is, why would they tend to have a frequency of 24 hours, a period of 24 hours? Right? Why don't they have all completely different frequencies and it's a complete mess? That's not what you see. You see this very strong 24-hour periodicity that is very pervasive. So we think that the reason why all these loops tend to have a 24-hour frequency is because of evolution. So you know these famous experiments or these famous accidents actually of bridges that were built without taking into consideration the frequencies at which they could resonate. And then when an army battalion walks on the bridge or there is a wind that excites the bridge at the right frequency, the bridge starts oscillating at very high amplitude and collapses. So this is just an illustration that physical objects tend to have intrinsic frequencies at which they vibrate. And the results I showed you is that the loops in the cell, they seem to have an intrinsic 24-hour frequency at which they vibrate. Why? I think the reason is evolution, because you have to realize that the world is very different during the day and during the night, right? The tides are different, the temperatures are different, the winds are different, the predators are different, etc. So it is extremely important and useful for a biological organism to be able to predict the day and night cycle. And if you ask how many times the Earth has gone through a day-night cycle since the origin of life, the answer is a very large number of times. You can do a back of the envelope calculation. It's 3.5 billion years, so 10 times 10 to the 9 times 365, you get 1.3 trillion times that the Earth has gone through a night-day cycle. The number actually is more like 2 trillion times because the day-night cycle is getting slower due to tidal effects. Hundreds of million years ago, the day-night cycle was 20 hours, and now it's 24 hours. So we know that it has been slowing down, and if you do a slight correction, you find a number that is closer to 2 trillion times. But the bottom line, it's a very, very large number of times that life has gone through this cycle. And then if you look at the first organisms, some of the first organisms 3.5 billion years ago, you had cyanobacteria, which are organisms that use photosynthesis. Now, if you use photosynthesis, you are entirely circadian by definition. You derive your energy from the light, right? That's what a photosynthetic organism is. So the first circuitry, the first molecular circuitry of life, 3.5 billion years ago, was completely circadian. And all the circuits we have today are derived from those circuits through this 2 trillion revolution of the Earth on its axis, right? So I think that's the explanation for having a lot of loops, a lot of oscillators inside cells that like to vibrate at a 24-hour periodicity. Okay? All right. But that doesn't tell us how a cell decides which oscillator to engage in a given situation, right? Imagine a cell, it may have 100,000 oscillators. They are coupled to each other. They may have frequencies that are close, periods that are close to 24 hours. But at the end, you only see 10% of molecular species oscillating under a given set of conditions. So there must be ways by which the system decides which genes should be expressed in a circadian fashion under a set of genetic, epigenetic, and environmental conditions. And so we're trying to make a theory of that, and you basically have two extreme cases. One is the case where you think that everything is controlled by the clock, the core clock, those 15 genes that I showed you. And it's the core clock that decides which other loops should be oscillating. Or at the other extreme, you have a completely self-organized system where all these oscillators are competing and cooperating, and somehow they find some kind of equilibrium. Okay? And the question is, where is biology operating in the spectrum? As you may guess, it is somewhere in between. But where in between? Okay. So to go beyond this, which I showed you, which is just the frequency at which you find a particular gene behaving in a periodic way across many experiments, what you have to do now is to look at pairs of genes, or more than that, and trying to look at infer circadian regulations between different genes, in particular between transcription factors and their targets. So what we do is we develop a method which we call the CRC methods, which is a way of establishing when, essentially we're trying to infer causality. We're trying to infer when is a transcription factor responsible for the oscillation of the transcript. Okay? So for that, we need to have at least three conditions. You need the transcription factor to be oscillating with a 24-hour period in that particular experiment. The target transcript has to be oscillating also with a 24-hour periodicity. You need to have, so both have to be oscillating. Of course, you need to have evidence that the transcription factor can control that transcript. So for that, you have to look in the promoter and look at whether there are binding sites for that particular transcript in a binding site for the transcription factors in the promoter of that particular transcript, so that you know that the transcription factor has the potential for controlling the transcript. And then the third condition is, ideally, they should be in phase, right? You would like the transcription factor binds to this transcript and they oscillate together, right? And ideally, they should be in phase. However, we know that there is a delay between the time the transcript reaches its maximum, for instance, the transcript of the transcription factor reaches its maximum, and the time the corresponding protein reaches its maximum. There are delays, so you have to take into account those delays. So we've been working on that. We have some experiments where we have measurements of both the mRNA and the protein levels, and so you can look at the lag between the mRNA and the protein level. To make a long story short, on average, it's on the order of 5 hours, 4 to 5 hours. And if you take plus or minus 2 hours, you get a good coverage of most of the cases. Right? So our three conditions to say that the transcription factor is responsible for the oscillating of the transcript is that they should both oscillate. The transcription factor should have binding sites in the promoter of the target transcript, and then they should have the right phase lag, on average 5 hours plus or minus 2 hours. Okay? So now we can look at all those experiments we have and put an arrow between a transcript and between a transcription factor and transcript when they satisfy these three conditions, which are suggestive of the transcription factor controlling the transcript in a circadian manner. And so you're getting a graph with all these arrows. It's a directed graph with arrows that you can get for specific experiments, or you can aggregate the graphs and get an aggregate view across all the experiments, the circadian experiments that you have. This is a table where we're looking at different tissues. So you see here, this is a combination of all tissues. This is just brain, skin, liver. And we're ranking transcription factors or RNA binding proteins by how often they are involved in a regulatory circadian edge. And again, just by doing these simple constructions, you find immediately a lot of interesting biology. You find transcription factors that seems to be very often involved in circadian control. And some of them are very often involved in circadian control only in brain or only in skin, etc., etc. So each row here, is something that you can either check whether it's reported in the literature or whether you have a new finding. And I won't go through each one of them, of course, but just to show you how these high throughput techniques can be applied and are very useful. The most interesting picture is perhaps this one, where you see, here is the core clock, and then you can look at all the genes that are a distance one from the core clock. So the core clock contains a lot of transcription factors, and you can look at all the genes that have binding sites in their promoter regions for the transcription factors that are in the core clock, and that are also circadian, and that have the right phase lag. And you see about 30, let's say 35% of genes, it depends on which experiments, satisfies these properties. So this tells you that the core clock directly controls about 35% of genes and enables their oscillation just by direct interaction, one step. If you go 2 steps away, you find about 70%, and 3 steps away about 82%. This is done in mouse. You can do the same thing in baboon. You get similar charts. So what is this telling us is that, remember this question of the spectrum, where are we on that spectrum? Is everything controlled by the clock, or is everything more chaotic and self-organized? And it's somewhere in between, where you see the clock playing an important role. You can think of this as being sort of a tree, where you have the core clock at the apex of the tree, then you have all the transcription factors at level 1, which are at distance 1 from the clock, distance 2 from the clock, distance 3 from the clock. And you can see here the proportions, and you can also look at the fan out of the transcript in this different level of the hierarchy, and you can see that the fan out decreases as you go down in the hierarchy. So at level 1, in this, if you look at the transcription factors that are in this section here, they have a lot of fan out, they have a large fan out, they can control a lot of transcript. When you go at this level, the fan out is reduced, and it's very small when you're at this level, it's 2 or 1, right? So you have this hierarchical organization. There is a certain number, a small fringe of genes here that never oscillate across all the experiments we have. And there are some housekeeping genes there, but most of them are actually olfactory GPCRs. So these are G protein coupled receptors. These are important receptor proteins. And we have about 2,000 of them that are just dedicated for olfaction. These are the proteins that are in the neurons in your olfactory epithelium. These are the proteins that bind to different odorants and allow you to recognize different odors, right? And there is no reason to believe that these should be produced in a regular, 24-hour, with a 24-hour periodicity. But all the rest of the transcriptome, again, we find that any transcript is capable of oscillating. Any one of the sort of remaining transcript is capable of oscillating in at least one set of conditions. So we have this hierarchical organization with the core clock at the apex, level 0, 35% of transcript and at level 1, etc., etc. The fan out decreases with distance. There is, of course, feedback between these levels, mostly between one level and the previous one. That's where we see the majority of the feedback. And most of the cellular reprogramming must occur at the clock and at the first level, because that's where you have the largest fan out. And there is a small set of transcript that seem to never oscillate, and it's primarily olfactory GPCRs. So here's the core clock. Here is the level 1, all the things that are at distance 1 of the core clock. And if you try to cluster them by functionality using things like David and cluster them by Go term, you find groups of functions that are quite interesting. For instance, you find a number, these are transcription factors involved in metabolism. These are the RNA binding proteins that I mentioned to you. There's a bunch of transcription factors that are involved with neuronal function. You have to realize that circadian rhythms are very important in the brain because, for instance, at night, you go in a very special state of sleep and dreaming and REM sleep, etc., which is very important, for instance, for storing memories. And then you have the cell cycle, which is what Indica was mentioning, which is also very important and coupled with the clock. In both ways, you have members of the core clock that are binding in the promoter region of cell cycle genes and vice versa. So that's the summary for the circadian portion of my talk. In any experiment, you have roughly 10% of genes that are oscillating in a circadian fashion. Genetic, epigenetic, or environmental changes can shift which genes are oscillating in a cell or in a tissue. There are a number of tools that allow you to analyze those oscillations. Circadiomic is a repository that is available on the web where we have aggregated several hundred such experiments. And by analyzing this data in a high throughput fashion, we're able to see the beginning of general organization of circadian oscillations, where you have a hierarchy of transcription factors that starts with the clock and has different levels, where the fan out of the transcription factors at the different levels tend to decrease, and where most of the reprogramming is done at the first level, one step away from the core clock. In time, we hope that these methods, this analysis, will have applications in precision medicine and other areas. We believe that it may be possible to develop better diagnostic methods by looking at oscillations and better ways to optimize drugs. You have to realize that it's very difficult to do these experiments in human. You can do in blood. We have a few experiments done on blood cell, but you cannot take biopsies of the liver every 2 hours just for the sake of understanding rhythmicity in the liver. It's not something you can do easily. So this is why all the experiments are done in mice, but of course then you have to extrapolate to humans, and that's not very easy. So that's the end of the circadian part of my talk. I don't know if there are any questions, otherwise I'll try to go over the neural network capacity portion of the talk. Yes?
Yeah, I have two questions. You built up some hierarchy of
Yes.
the regulatory network, and my first question is, how did you obtain the network? Is it really some physical regulatory network, or is it kind of just inferred regulatory network? This is my first question.
Oh, it's all inferred from the data. So basically, think about transcription factors. Those are the key players, right? You have about 2,000 transcription factors. 10 of them are in the core clock. And then you have the first level, all the transcription factors that are distant to one from the core clock, because they have core clock binding site in their promoter regions. And then you can define distance 2, distance 3, right? And so that's how we build the hierarchy.
Okay. And the second
And then we look at which transcription factor are oscillating in a given experiments and their connections, using the methods I described, okay?
And the second question is, you said that if you get the intersection of the all genes, then around 2% of genes, they are actually commonly show the rhythmicity across the conditions. But if you have the union, then around 95%, I remember. So did you get any interesting visuals about what these 2% genes, they are always oscillating?
So the 2%, the 2% is just in one experiment. So if you do 2 types of diet, typically you're going to get about 2% in the intersection. Right, that's 400. Right? So the 2% is different across different intersections, but if you take what is common to all of them, you get the 4 clock, which is about 15 genes. Okay? All right, let me try in 10 minutes to shift gears and tell you a little bit about neural network capacity. So very often in machine learning, what you have is a class of hypotheses or a class of functions, and there is a function that you want to learn. Very often you know this function only through examples, and basically learning can be viewed as starting from a function in your class. You have some kind of learning process that tries to find the best function, the best approximation to the function h within your class. Very often h is not inside the class of hypotheses or approximating functions that you have, right? And ideally what we would like to do is to take a neural network with 5 layers of size 300 and say, can we say something about the capacity of this ball here, right? What kind of functions can you approximate if you choose this particular family of hypotheses? So that's a very difficult question. Nobody has been able to make much progress. I'm going to slightly simplify the question by trying to measure just the size of this thing here. And because, in general, you have a lot of functions in this space, in this ball, I'm going to take the logarithm base 2 of this volume. if you're doing things in a continuous manner, you may have to define what you mean exactly by volume. You will have to put some kind of measure. But I'm going to work in the discrete case. So for me, I'm just going to count how many functions are in this space here, and then take the logarithm base 2 of the total number of functions. Okay? And the idea is that if you have a class of approximator that has a bigger volume, in general, everything else being equal, it is a better class of approximator than a class that has a much smaller volume. All right, so we're interested in neural networks, so we're going to look at very simple neurons. You're probably familiar with these neurons if you're doing machine learning or neural networks, where you have a bunch of inputs, a bunch of weights. You take the weighted average of your inputs, and then you pass it through a nonlinear function. And the nonlinear function I'm going to use is the sign. So I'm just taking the sign of the sum of the WIXI. That's called the threshold gate, if you are an electrical engineer, right? And a slight variation on that, instead of taking the sign of a linear of a linear combination, you can have a polynomial in your input variables with some coefficients and take the sign, right? So this is a special case when the degree is 1. We'll look at cases where the degree is 2, 3, etc. And you can have a number of variations. You can constrain the weight to be, for instance, binary or just positive if you want to think about, say, an excitatory neuron where all the connections are positive. You can put restrictions on the weights. Okay? But we're going to look at this binary, these Boolean functions, threshold gates, Boolean function. And we're going to ask again, what is the log 2 of the total number of functions that can be implemented as you vary the weights? So imagine first that you look at Boolean functions, and you ask which ones can be implemented by a linear threshold gate. Okay, so it's equivalent to say which Boolean function are linearly separable, right? So think about the hypercube, right? -1 + 1 to the power n. And imagine you color the corners red and blue, and you're asking how many colorings can be separated by a hyperplane, right? Obviously, if the coloring is random, you're not going to be able to separate it with an hyperplane. There is only a very small number of colorings that is separable, that is linearly separable. That's what we're trying to compute, right? The number of Boolean functions that are linear separable, the number of Boolean functions that can be implemented by a linear threshold gate. And then you can do the same thing with a polynomial threshold gate, okay? So here's the space of all Boolean functions of n variable. You know that it's 2 to the n. That's the total number of functions. So if you take the logarithm, it's just 2 to the n. And then you have those that can be implemented that are linearly separable. For instance, the AND function is linearly separable, the OR function is linearly separable, etc. There's a bunch of functions that are known to be separable, but there is also functions that are not separable. And in fact, most of them are not separable. For instance, exclusive OR or parity is not linearly separable, right? So how many Boolean functions are linearly separable? This is a problem that has a long history. When you take the logarithm of the number of linear threshold gates, you find that the logarithm is upper bounded by n squared. This is a theorem that was proved by Carver and others around 1965. There's a lower bound that was proved by Muroga around the same time, which is a constructive lower bound. There is a way of constructing a subfamily of threshold function that gives you this lower bound of c to the n squared. Again, this is the logarithm. C to the n squared is the logarithm of the number of the linear threshold function. And in this construction, c was strictly less than 1. It was 0.5 actually. So that gives you a gap between the upper bound n squared and the lower bound 0.5 n squared. And the problem was settled in '89 by a Russian mathematician Zuev, who proved that the upper bound is actually the right order. So the answer is that the logarithm base 2 of the number of Boolean function that is linearly separable is of order n squared when you have n input variables. Now, if you look at polynomial threshold functions, you have a similar situation. So I'm going to look at polynomial threshold function. In 88, I proved this upper bound. So now you have polynomial threshold function of degree d. Before it was just d equal 1. Now I'm looking at general d. So for instance, you have a quadratic form, and you take the sine of the quadratic form, and you're using that to partition points in the hypercube, or a cubic form, etc. So this was the upper bound known in the 80s. Michael Sachs proved a lower bound of n choose d plus 1, which leaves a gap between these two, because the denominator here is d plus 1. This is essentially n to the d plus 1 divided by d plus 1 factorial, and this is d factorial. So there is a gap between d plus 1 factorial and d factorial. And recently, Ronan Vershinin and I, we proved the same thing holds for degree d, which is again that the upper bound gives you the right order. So if you look at the logarithm base 2 of the number of polynomial threshold function of degree d on the hypercube, you find that it's on the order of n d plus 1 over d factorial. So that solved the problem for a single neuron. So if you have a single neuron, whether it's a linear threshold gate or polynomial threshold gate, we know how many such objects there are. Of course, you want to go to circuits. And, well, before I get there, let me show you this picture. So this shows you how the Boolean functions are stratified. If you have degree equal 1, you're looking at the linear threshold function. As I told you, it's order n squared. If you increase, you go to the degree 2, you find n cubed over 2. You're going to find nd plus 1 over d factorial if you go to degree d. And then if you play the game of looking at subsets of functions, for instance, linear threshold function with only binary weights where the w's are just plus 1 or minus 1, or in that case, the capacity goes down to n. From n squared, it drops to n. So restricting the weights to be plus or minus 1 is very restrictive. You lose one order of 1 power if you do that. On the other hand, if you look at linear threshold function where the weights are constrained to be positive, you don't lose that much. You go from n squared to n squared minus n. Those of you who like to play with neural network, you should know about ReLU function, rectified linear units. You can show that it's n squared plus n. So it's slightly more powerful than linear threshold gates, but not by much. Okay, all this is for single neurons, single threshold gates. Of course, you would like to go to networks, and this is what we're working on. But you can prove a very simple theorem that if you have a set of n threshold gates that are fully interconnected to each other, then the capacity is n cubed. Essentially, because each one of them has capacity n squared, and you have n of them, you get n cubed, and you can see that this is actually the right order. So a fully interconnected recursive circuit of n linear threshold gates has capacity n cubed. It can implement up to 2 to the n cubed different functions. And we're extending this to layered feed-forward networks, so we can compute the capacity, for instance, of this circuit, which has this kind of expression. But I don't have time to go into details. But just to give you the general idea, you can define, provide a very simple but precise definition of capacity as being the log base 2 of the number of functions that can be implemented by a particular class of, in this case, of neural networks, but it could be other things. Capacity of linear threshold gates or polynomial threshold gate. We have precise estimates of those quantities. You can compute capacity in special cases where the neurons have binary weights or weights that are constrained to be positive. A fully connected network of n such neurons has capacity n cubed. More generally, the capacity is going to be, at least the dominant terms of the capacity are going to be a polynomial in the relevant variables of the circuit, which are the number of neurons, the fan-in of the neurons, etc. So in general, you're going to have a polynomial in those variables, relatively low degree polynomial, and it can be estimated, at least in the case of a fully connected network and a two-layer network, and we're currently working on multi-layer feed-forward networks. Any questions on the second part of my talk? Yes?
I have two questions. So one is about the capacity, and can you tell us a little bit whether larger capacity means the model can learn an object from a larger feature space? I just don't know what really capacity is. And the second one is this: the model with larger capacity always better, perform better than smaller capacity.
All right, so your first question, it's not the number of features. Number of features is sort of the input, okay? So you could have an input of size n, but then you could have a neural network with one layer or a neural network with 5 layers. And you could ask, do they have different capacity? So that's...
But this larger capacity can learn more, like things from higher...
So is it better to have larger capacity? That's your question. Everything else being equal, the answer is yes, because you can cover more functions, you can approximate more things. Okay? So everything else being equal. Of course, if you have a class of functions or a class of hypotheses, the notion of capacity I'm using now in a sort of intuitive sense is much more subtle than that, because what you really want to know is what kinds of functions it can approximate. So it's not just about the size, but where is it in space, right? So what you really want, when you want to characterize the capacity of a class of functions, you want to know, how big it is and where is it in space and what it can approximate. So it's a complex notion and it has different facets. Here I'm taking only one aspect, which is how big it is. But in a specific case, you could have something very big and something very small, and the small one is better because it is in the right position in the space you're interested in.
So we still need to try, like try different ones.
Yes.
I have a question. Part one, I don't get the purpose of that automated networks. Is it for calculating
the autoencoder I showed briefly, the goal is this. If you do a gene expression experiment, usually it's done at a single time point, right? You measure 20,000 genes at a single time point. And you give me that data. I would like to know when did you take the measurement. Right? I want to know what was the time at which you take those 20,000 measurements. Because in reality, it's a bad idea to take measurements only at one time point because of all these circadian oscillations. You should have taken many, many experiments, maybe every 4 hours, to really get a good picture of what was going on in the biology you were looking at. But you didn't. It's expensive. You did it only at one time point, and you didn't record the time. The question is, can I just look at your 20,000 measurement and guess what was the time? So to do that, we have this autoencoder network where you put the 20,000 measurements in the input. Okay? You have a bottleneck which has a sine and cosine function. So it's trying to predict what's the phase. And then in the output, you try to reconstruct your input. So basically, you're trying to find what are the two variables that are the most important in order to reconstruct your output from the input, and you do this for many such experiments. You go to the Geo database that has a lot of gene expression measurements, a lot of them at single time points, so you have a lot of training data for training this autoencoding.
You estimate the capacity, but you use neural network, different layers of neural network. Did you compare like capacity between this neural network and some other machine learning algorithm, like
Not yet.
CDM, decision tree?
That's a good question. We haven't done that. The reason we started this, we wanted to compare deep versus shallow networks with the same number of parameters. So that's the main case we were focused in. But of course, you could do those other problems are also interesting.
Thank you very much. We will have a